Okay, so after you send your sample to a facility, by the time you get the sequencing, it's a FASTQ data. And if this is a paired end sequencing data, most of the RNA-seq nowadays, you get a paired end sequencing. For other applications, you might get single end. For RNA, it's 2 files, and it's line by line. It's the first read and the second read of this fragment. It's not always, it's not the case that the first read has to be always on the plus strand and the second read has to be on the negative strand. It could be the other way around. It doesn't mean, well, half-half. Okay, so you usually get 2 reads. And so the very first step, we are looking at, in this case, a simple case. You are sequencing some samples from human mouse. The genome has been very well assembled and annotated. And so the very first step is aligning the read to the genome. So yeah, so in the last lecture in high throughput sequencing, we explained alignment. We discussed the BWA and the bow tie, and both use this Burroughs-Wheeler transformation for compression and for the LF mapping for the finding the alignment. So the question is, do we want to use BWA or bow tie to align the RNA into the genome? Do we want to do that? So remember, this is the genome, and when you have a gene, there are exons and then introns and exons and introns. And so if you were to only align based on a genome using algorithms such as BWA and bow tie, it will be able to align the reads that really fall within an exon, because it can really find a location. However, when you have a read that cross a exon junction from one exon, skip to the next exon, regular algorithm like BWA and bow tie would not know what to do with it, and so it will throw it away. And so therefore, when you have RNA-seq, we want to use something called the splice-aware aligners. The very first one is Top Hat, and the later generation from the same group is called HiSet. And recently there is also algorithm using suffix tree. So these are BW approach because Top Hat is built on bow tie. Later on, there is also algorithm called STAR, and in the homework, you can try out STAR for alignment. I can just roughly show you how this splice-aware alignment does. So you still need the genome index file. The transcript annotation file is optional, but it's not required. You don't have to know the gene annotation. As long as you know the genome, it's okay. And so this is the rough idea of Top Hat. So at that time, the reads are quite short. Top Hat, at that time, people were reading 25 or 50 base pair read. And so what they do is they first try to map any reads to a full exon. So initially, it was like a bow tie. You just kind of, or BW approach, you map the reads that you can map. And so then once the read is mapped there, you know roughly there is an exon in there. And then based on the known, if there are enough reads in here, you can create an exon. And there are previous kind of from gene annotation, people know that usually right after the end of an exon, you have a GT, and the beginning or right before an exon, you have an AG. This is called the splice donor and acceptors. And so that's the signal where the 2 exons can be, or the introns can be spliced out and exon can be joined together. And so after you use the reads to define the exons, then you can create these junctions. Then kind of create a new index and then map the unmapped reads to these junctions. Then you can, hopefully get this in a couple of rounds, then you will get all the transcript mapped to the junctions. So this is the rough idea of Top Hat. Recently, the sequencing reads are getting longer and longer. As we mentioned, we're getting 100 base pair reads, and sometimes you have paired end reads, right? So these are pretty long. And the average length of an exon is about 150-ish. And so you could have shorter exons when a single read can cross 3 or 4 exons even. And so that could really make it hard because originally you could have a full read mapped to a single exon. And now a lot of the single read will cross junctions. And so now with longer reads, sometimes you can break it up into shorter reads and see, can I map the first 25, second 25, and so on. And then use those mapped 25 base pair to try to do extension. But you can get the rough idea. You map what you can, then you try to extend, you create the junctions, and then try to, and sometimes you will see that there are junctions that people weren't aware of before. So they create new junctions. And so after you use either HiStat, nowadays people either use HiStat or use STAR. STAR is considered currently the most accurate one, but although it's a little bit slower. After you finish the read mapping, you will get a BAM file, and because it's PE, I'm showing you an example of this is a one strand, sorry, the one end of the read. This is another end of the read. So the first column is still the ID. So this one is kind of the, remember last time we did the single read, and now with a pair read, this, if you convert this number into a binary, there is a table you can look up, is this a unique read, or is like mapped to multiple locations and things like that. The first bit always indicate that this is actually a paired end sample. And so all the paired end samples will have odd number in here. You can convert this into kind of a bit string, then based on the first bit, you will know that it's a single, or it's a paired end read, which means that you will have, this is a mate pair. Mate pair means this is one fragment, one read and another read. They will have the same ID. They may not be adjacent lines in your read file, in your final file, but based on the ID, you will know that they are from the same mate pair. And so this is actually the important part that you kind of, well, so this is saying that this is mapped to chromosome 497 to 73 location for this read. And then what it has is that there is a 27 base pair match, and then there is a 1099 base pair gap, and then another 48 base pair map. So that means in this read, it actually crossed an intron of 1,099 base pair long. So the left side, there's 27 base pair. On the right side, there's 48 base pair. But in the middle, there's a big gap from an exon, right? And then this is the location of the other read. And so the two read pair, the mate pair is 4,970 base pair apart. So that's the insert size. From the beginning of this to the beginning of the other side, they span actually pretty long. On the human genome, it's not because of the RNA is that long, it's on the human DNA genome, they are almost 5 kb apart. Okay, and then here is the sequence, and here's the quality score, and this read is like how many mismatch do you see in this location. And if you look at the reverse strand, this is also chromosome, it's mapped to this location. You can see this location is the same as this location. And here, there is initially a 19 base pair map. And then this is the other read now, because this is, what maybe it covers 2 exons. This is the other read. And then this thing cross over an intron, which is 566 nucleotide. And then another 56 base pair match. And then this is the other read, the first read that we saw, right? And then that distance is a minus sign because it's from this region back, right? And then this one also, this has the direction of the transcript that's underlying this. So it could be that the gene is on this strand. It could also be on the opposite strand, okay? And so roughly you will get this information. And with the read mapping, we can now also do additional quality control. And there are a number of these RNA-seq quality control algorithms. For example, RCQC. So it has a number of functions. It will generate a big figure of all the many figures for your sample, but this is a few of the important things. The first is you do fast QC just to make sure the read quality is okay. Remember, we want to see, this is actually not a very good sample, but you want to see that the read quality is still very good as you go longer and longer into the read, right? So the beginning of the read is good, and as you sequence longer and longer, it might fall off. This is just one example. You want this to be very high throughout, or as long as possible. And the second part is the nucleotide composition. We mentioned that sometimes the beginning of the read might be kind of crazy. And in that case, you could trim out the first few bases of every read. And actually nowadays, I think sequence aligners automatically deal with this situation. You don't have to even worry. This is partly due to the random priming step in the RNA. When you are making the RNA sequencing library, the priming can have this issue. But this is an experimental step, and the alignment will deal with this. And so sometimes the final alignment you may not have. If you sequence exactly 150 base pair read, you may not actually get exactly a mapping of 150. You might just map 100, which is still very good. Another thing is you want to kind of roughly look at your insert size. Because you are doing a paired end sequencing, usually you want to see what is kind of in between. This is not on the genome. This is on the RNA. You're just trying to estimate. Remember the initial RNA, we fragment them to make them shorter from a few kb to maybe a few hundred base pair. And you roughly want to know how long is my fragment when I sequence them. This is by looking at how far apart they are on the final transcript. And I give you a rough distribution. You don't want this to be too long or too short, but just give you some idea of this. And also another important readout is how many of my reads really landed in exons. In this case, this is not the percentage, but the count, or how many reads per kb. But you want most of your reads to land in coding exons, and maybe occasionally some non-coding exons of 3' UTRs, 5' UTRs. And then the rest, these are really mapped to potentially introns or outside of the gene or some novel transcript. Yes, you might have some novel transcript, but they shouldn't be predominant in your sample. Otherwise, there's something wrong with the sample processing. If you do a poly A transcript and seeing most of the read land in exons, that's definitely a good thing. You can also look at the splice junctions. If one read is split, half of it mapped to one exon, the other half mapped to another exon. By the way, so RCQC, you do need to use existing transcript annotation, because there are standard gene and transcript annotations now. So you want to ask, out of all the splice junctions we see, how many percentage are known before and how many are novel, completely novel or partial novel. So you want to see a little bit, but probably not too much. Otherwise, maybe there's something wrong with the sample preparation. And a very important thing, if you have a sample that you suspect might be degraded, is this transcript integrity number. You will see a figure like this, which is kind of the read coverage on each gene. You basically stretch and squeeze all the genes so they are all the same length. And you look at each transcript, like first of all, you look at each transcript, you ask, if this gene, you can imagine if it's expressed a lot, there will be some reads that cover the beginning, middle, and towards the end of the transcript. You will have a fairly even coverage like this. You may not have the very beginning of the transcript, because that would require the random priming of the sequence. To generate the cDNA, you need some random priming. Sometimes that could be hard. And also, if the fragment is very short, you may not really make it in your sequencing library as well. So you may or may not have good coverage in the very beginning and the very end, but most of the gene body, you should have a good coverage. So TIN is calculated on every transcript in that is detected in the cell, but MedTIN is looking at all the transcripts and look at their kind of average or the median of all the TIN scores. And so you can see this is one sample. Well, these two are kind of essentially the same. We have multiple samples. If you have a sample like this, it means it's a good quality sample because most of the transcript, you have a good coverage throughout the gene. But if you have a sample like this, where you only see the 3' end of the transcript on the genes, that indicates the RNA has been pretty badly degraded. And so when you have a teen score less than 50%, that's not a really good sample. And you may or may not even want to use the result. You will get some sequencing, but you probably don't want to use it. Occasionally, if this curve becomes really jagged and crazy, you also have to be careful whether there's something wrong with the sample quality. Okay. Question about alignment and quality control. Okay.